Okay, so is it visible to all?
Yes, sir. It's
The second part of your syllabus is Evolutionary Biology Part 2, and under this is the second unit. Under this, the first part, we have to discuss about the three domain system, the evolution of organism. What actually it explains? Evolution explains how the organism change with the course of time. So in this format, if we look into the back, if we look into the evolutionary time period, we can understand that uh, the organism first started in aquatic medium, and those organisms are unicellular organism. In the previous class, we have discussed that how unicellular organism or very simple form of organism were formed by the process of chemogeny and biogeny. In RNA world hypothesis, we have discussed what is chemogeny and what is biogeny. On that basis, this domain system means whatever living things are present on the surface of Earth, they belong to which category? For identification purpose, we have to segregate the organism based on certain properties. So on that basis, based on certain properties, all the organisms are categorized into this three domain system. Okay. Under this, we have to discuss, or we are going to discuss about the universal common ancestor, tree of life, three domain concept of living kingdom. Okay. So first come to this universal common ancestor and tree of life. So what is this universal common ancestor? It is it refers to actually the last universal common ancestor. Universal common ancestor, generally it is written, but it is abbreviated as LUCA. LUCA means last universal common ancestor, from which all life on earth evolved. Before some time, I have told you that all the life forms, they form from the complex chemical substances. Those complex chemical substances, they form the coarservate microsphere. After coarservate microsphere, they are converted to life form when they engulf, when they ingested the ribonucleic acid, the deoxyribonucleic acid, they converted to the living cells. Okay. So these are the concepts we are going to discuss. So this universal common ancestor means last universal common ancestor from which we all are derived. Not only we, all the organisms, all the complex organisms, simple organism, everything derived from this. So Luca is hypothesized to have existed around 3.5 to 4 billion years ago. It means that life form, Luca means origin of life. How life originated, or when life originated. This 3.5 to 4 billion years ago is the origin of lifetime. That means before that, there was no life. After that, life was formed on the surface of Earth. Okay. So this all about the universal common ancestor. This is the idea about the universal common ancestor. Now come to next part. That is the tree of life concept. Tree of life concept explains what? It's a graphical presentation, metaphor. Okay. It's a graphical presentation or diagrammatic presentation which represents the evolutionary relationship among all the living organisms. If we draw the life form, just look at this picture. Here, this is the Luca, last universal common ancestor. Mark the picture very carefully. How life was originated. This point is the origin point of life. After this, life diversification. That means towards the origin, diversification or biodiversity is very less. If we move far from the origin, far from the beginning, then we got more and more diversified life. Okay, so this means the diagrammatic arrangement of different form of life based on their origin, based on their different properties. How life originated, or from where, or from which point life gets separated from its origin. Okay, that concept is the tree of life concept. Now, the molecular evidence explains us the tree of life is largely supported by molecular data, especially comparison of ribosomal RNA. If we compare these organisms, let's say this is the organism Luca. Again, this Luca was there, first universal common ancestor. Luca is the last universal common ancestor. Fuca is the first universal common ancestor. That means that was considered as the common ancestor before the Luca. Okay, so we don't need to discuss on that part, but these are the segregation. From this point, life sustaining material are present on the surface of Earth. Okay, this Luca, last universal common ancestor, was actually the life form, and those life form, they deviate from each other, from the origin, and make these three domains: bacterial domain, archaeal domain, and eukaryotic domain. This is a three domain classification by Carl Uge. In three domain classification, all we know that how many kingdoms are there? Six kingdoms. All we know that six kingdom classification by R.H. Whitaker. So these six kingdom classifications, that means all the organisms are coming under this six kingdom classification, and this six kingdom classification are coming under this three domain due to their property. And this three domain system based on, that means in which property we can segregate them, we can isolate them from another. That is the supporting data, and for this the supporting data is the molecular data, particularly the genetic material. Here the genetic material, when Carl Uge segregated this property, he used genetic material as RNA, ribonucleic acid. Okay, so these are the very beautiful diagram of this Luca. From this past universal common ancestor, certain ancient cells were derived, but those ancient cells, with due course of time, they unable to adapt themselves. They were extinct. But the last universal common ancestor, they adapt themselves with the changing environment. They adapt their genetic composition, genetic material with the changing environment. And for that property, they survive, and in different environment, due to different adaptability, they isolated from each other. They derived from the Luca and segregated, bifurcated to different form due to their different adaptability characteristic. Okay, and the last part is the evolutionary relationship That means the tree of life shows that eukaryotes, bacteria, and archaea have diverged from a common ancestor. On that basis, we can say that they all belong to the common ancestor. They all derive from the common ancestor. But with course of time, they segregated from each other due to different habitat, due to different food source, due to different environmental challenge, climatic condition, everything. Okay. Some of them they prefer the aquatic medium. Some of them they prefer the uh, terrestrial condition. Some of them prefer the swampy lands like amphibians. So based on that characteristic also, we can classify the organisms. So on the broad sense, on the broad classification, they are classified into three domain system. And this three domain system, just look at here, the bacterial domain, they the bacteria, first kingdom, the bacteria coming under the bacterial domain. The archaeal domain is the archaea. Eukarya domain, they are the protist, plant, fungi, animals, all these are coming under the eukaryote domain. Okay, so these are basic classification, phylogenetic classification, tree arrangement of different life forms. All of you clear or any doubt? Am I audible to all?
Yes, sir. Clear.
All of you clear about tree domain
Sir, I have
system?
a question.
Yes.
Sir, why rRNA is considered for a phylogenetic tree?
Why?
Why
Why?
rRNA is considered to evaluate the phylogenetic tree? Why not DNA or RNA?
Okay. A very good question. Why not DNA or RNA? Not only rRNA, particularly the most of the case RNA. Okay. And why we mention the rRNA? Because us or the primitive cells, they have ribosomal RNA. If the primitive cells or the simplest cell have the ribosomal RNA, that means that ribosomal RNA was firstly evolved in comparison to other group of RNA and DNA. In yesterday's class, we have discussed the very simplest structure, and it was hypothesized that the simplest structure was first evolved, not the complex structure. Okay. And the second one is why RNA? Because the genetic material generally they do not change. If they change, the genetic change in genetic material occurs, that means there is segregation, there is separation of organism, there is isolation of organism from the original organism. That's why scientists, particularly we mentioned Carl Uj, he emphasized on ribosomal RNA sequence. Okay.
Yes, sir.
So this is the history of three domain system proposed by Carl Uss in 1990. Based on differences in ribosomal RNA sequence, this system divides life into three broad domains. Already we have discussed. Okay, so these are certain examples of archaea, methanogens, halophils, bacteria, cyanobacteria, eubacteria, eukarya, all the eukaryotes, animalia, plantae, all these are the different examples of these three domain system. Now come to the characteristic of each domain. The bacteria They are prokaryotic microorganism without membrane-bound organelles. All we know that prokaryotic means they do not have proper nuclear membrane. Prokaryon, primitive nucleus. That means nuclear membrane was not developed at that organism or the phase of the organism. Okay. Not only the nuclear membrane, many cell organellar membrane also not developed. That's why we can say that they lack of many cell organelles, the primitive form, the bacterial form, the bacterial domain, or the organisms are coming under the eubacterial domain. They lack this form, membrane-bound organellar form. Cell walls contain peptidoglycan. Circular DNA was present, but not covered. Circular DNA where present is the space of nucleoid, but that is not considered as nucleus. No histone associated with DNA. They reproduce mostly by asexual manner, but in future sense or in future time, some of them they develop simple sexual reproduction. Okay, but most of the organism they reproduce asexually in the primitive time, and it includes diverse group of organisms, cyanobacteria, proteobacteria. Already we have discussed this all about the characteristic of eubacterial domain. Then come to the archibacterial domain. They are also prokaryotic, but genetically distinct from the bacteria. That means certain genetic changes are there. That's why, based on that property, based on the genetic material property, the segregation was there. Tree was, the branches are different from the eubacterial part. The cell membrane contain lipid. Okay. Cell wall lack peptidoglycan. In the previous form, the cell wall contain peptidoglycan. In the archaeal form, the cell wall lack peptidoglycan. Some have pseudopeptidoglycan. Okay. They prefer extreme condition. They can survive in extreme condition like high temperature, and they more closely to eukaryotes. That is the most important point. Just mark at the last point. Okay. We'll get some questions from this also. Why eukarya is more related? Very short question, 2 mark question, subjective question, that which domain is very close to the eukaryotic domain and why? This is the point. This archaeal domain are more closely associated to eukaryotic domain. Okay. Why? Because the property of the genetic material, like replication, gene replication. Okay. So this is the answer, that archaea is more close to the eukarya in comparison to bacteria. So we can say that this is the intermediate stage between these two, bacteria and eukarya. Bacteria is the oldest one, eukarya was the recent one, the intermediate one is the archaea. Okay. Then eukarya, organisms with true nucleus, that means nuclear membrane was distinct. It includes the animals, the plants, the fungi, the protists. Linear chromosome with histone proteins are there. Reduce both sexual, reproduce the sexually as well as asexually. Okay. Greater structural complexity than prokaryotes. So these are basic properties. All we know. These are 3 domain system and their basic characteristics. This
So
is
what is the difference between bacteria and eubacteria?
what is
The difference between bacteria and eubacteria.
bacteria and eubacteria.
Yes, sir.
Uh, actually, the bacteria is the general term, okay, that we use for the largest group. On the other hand, eubacteria is the true bacteria. Okay, there is no big difference between these two. But when we are saying bacteria, it's a big term. Eubacteria means we also can incorporate certain ancient organism like bacteria. But eubacteria means the organism having proper bacterial characteristics. Okay.
Yes, sir.
In that sense, we can say that the general group is considered as bacteria, but eubacteria is a specific group, or true bacteria is a specific group. Now come to the distinctions between domains. The first distinction is the genetic differences that explains us what? That explains the primary division between these domains comes from molecular difference, particularly in ribosomal RNA, DNA replication mechanism, and protein synthesis machinery. Okay, in this three form, these are the basic property. Particularly, scientists use this property. What is the coding pattern of ribosomal RNA? What is the coding pattern of DNA? How they are related? How they differ from each other? What is the mechanism of protein synthesis? Sorry for interruption. So we are at the position.
No.
Am I audible now?
Yes, sir.
Yes,
Sorry
sir.
for the interruption to whole class.
Yes, sir.
Uh, so our point of discussion was how these three domains or on which base, on which property we can classify these domains. Okay. So to classify these domains, scientists generally prefer to analyze their RNA, their DNA replication mechanism, the protein synthesis machinery, the central dogma technique, how DNA, they form RNA, they form protein, Okay, so these are basic property they use. The metabolic differences explains that is the process of metabolism in different domains. Not all the process of metabolism, that is the mode of ingestion, mode of digestion, mode of egestion, everything are coming under the process of metabolism. The respiration, the reproduction, everything, every process is different from each other. On that basis also, we can segregate these domains. And lastly, the cell structure is another important property. Uh, we all know that the prokaryotes, particularly bacteria and archaea, they lack nucleus. But the eukaryotes, they have nucleus. They have well-developed nucleus. In most of the bacterial cell, archaeal cell, they have lack membrane-bound organelles. But eukarya, they have membrane-bound organelles. Okay, so these are the basic properties scientists generally use to segregate these domains, three domains of life. Okay, then come to the importance of the three domain. Why we discuss about this three domain system? The three domain system, first evolutionary importance explains that the three domain system provides insight into the evolutionary history of life on Earth. Okay, that means which organism is coming from which organism. That means what is the proper pure ancestor of that organism. Okay, the organism, if genetically equal, similar, then we can say that this is the ancestor for this particular kind of organism. That is the evolutionary significance, evolutionary importance of three domain system or tree of life. Then systematic and taxonomy, which reshaped biological classification, challenging previous two kingdom and five kingdom system by introducing domain that represent fundamental evolutionary lines. That means how to classify these organisms. The groupism, yes, we are human being. We are coming under eukaryote. This is the plant on which basis we can classify. We can make a systemic study, a taxonomic analysis of that particular organism. When we are talking about the organism, that means we are talking about their name, their group, from which group they belong to, from which kingdom they belong to, from which phylum they belong to, from which order they belong to, everything. So this is systematic and taxonomic importance, and it is also helpful for modern research also. That means the system has been the system helps in which manner? The system helps in knowing the origin or pattern of life, how it originated. What is the diversity among the organism and how organism is different from another group. Okay, evolutionary process, that means different life form, how sequentially or in sequence manner they evolved from each other. So these are basic importance of this three domain system. Okay, and the last part of this chapter is the evolutionary significance of archaea and eukarya. As your syllabus concerned, the evolutionary significance of archaea and eukarya. They resemble, archaea mostly resemble to the eukarya. Already we have discussed. They share more similarities in their genetic and cellular process compared to the bacteria on the cell membrane structure, cell wall structure, different cellular activities. Okay, so genetic characteristic and uh, cellular structure on that basis we can segregate them, or we can make a systemic arrangement of these two domains. Okay, the evolutionary split between these domains represents a major transition in the history of life, with archaea likely giving rise to eukaryotic cells through symbiotic events such as endosymbiosis of mitochondria and chloroplast. That means how the eukarya was formed from the simple organism to the complex. That means when there was no eukarya, archaea was there, but on the dependency or the dependency between the organism, they help in the formation of eukaryotic group. Okay, that was explained in very well manner by this theory, which is known as endosymbiotic theory. And the best examples we have is the presence of mitochondria and chloroplast in eukaryotic cell. Okay, so what endosymbiotic theory explains? The endosymbiotic theory explains the origin of mitochondria and chloroplast. Okay, here it is written that the symbiotic relationship. What does it mean? Symbiotic relationship means both are interdependent. Both are interdependent without harming to others. That type of relationship is known as symbiotic relationship. So endosymbiotic theory explains that eukaryotic cell, when engulfed an aerobic prokaryotes means those group of prokaryotes, they use oxygen for the purpose of oxidation. That is known as aerobic prokaryote. When the food shortage was there or due to any mechanism, if that small aerobic prokaryote was ingested into the big cell, eukaryotic cell, and they become, they develop a relationship, symbiotic relationship, that the big cell, if this is the cell, just look at this picture. This is the big cell, which was not considered primitively as eukaryotic cell. That might be bacterial cell, that might be the archaeal cell. But here it is one non-eukaryotic cell, it is also another non-eukaryotic cell. Both are independently survived in the primitive environment. Okay, no interconnection between these two. I'm just explaining the endosymbiotic theory, very excellent theory on the basis of uh, for the evolution of eukaryotes, how eukaryotes evolved. This theory is still dead, well accepted theory. So due to any force, any environmental pressure, when these two cells come in contact with each other, and due to any property of the bigger cell, the smaller cell, which was aerobic, they ingested into the bigger cell. Now, two things might happen. Either the bigger cell completely digests the smaller one, or the bigger cell just use the smaller one. The second thing happened in the previous environment, in the primitive environment, according to endosymbiotic
Sir,
theory,
slides are
that
not visible.
the bigger cell,
Sir, it is visible.
it's,
It is visible.
Okay, okay.
I think you have certain network issue. That's why not visible to you
No,
particularly.
now it is visible.
Okay. Am I audible when I'm explaining endosymbiotic theory? Am I audible to all?
Yes, sir.
Yes, sir.
Okay. So the bigger cell make an interrelationship with the smaller cell. That aerobic in the aerobic prokaryotes, they have the capability to generate their energy. But this cell, didn't have the capability to generate their energy. So they make a symbiotic relationship that the big cell provide habitat or space or protection to the small cell, and the small cell provide energy to the large one. Now this become the eukaryotic cell, which was not previously considered was eukaryotic cell. In the future time, it was considered as eukaryotic cell. Okay. In the same manner, chloroplast also ingested inside the cell. So this endosymbiotic theory explains how eukaryotic cells evolved from prokaryotic cell. This is the basic explanation. If we go for the key points of this endosymbiotic theory, it explains that smaller cell gave the host, or the larger one become the host for the smaller. In return, the smaller one provide energy to the larger one. Okay, so these are the basic principle of this endosymbiotic theory. Over time, the two become completely dependent. That means the larger one and the smaller one started living inside the bigger cell in a mutualistic relationship, okay, or symbiotic relationship. This is the phenomenon. That's why now we can uh, we have some proof that this might happen, because when we are considered about chloroplast or mitochondria, they are present inside prokaryotic cell, but they have their own genetic material. Prokaryotic cell have genetic material in the nucleus, but apart from that genetic material, they have other genetic materials which are present in chloroplast and mitochondria. This is one of the important proofs that might happen. Endosymbiotic theory might happen. Okay.
Sir, can you please explain the last paragraph of this slide? The endosymbiotic theory is a class of hypothesis from that to up to the last microbial cell. I want to know the meaning of descendant of endosymbionts.
Okay. Your doubt is descendant.
Yes, sir.
Okay. Descendants of endosymbionts. The paragraph explains the endosymbiotic theory is a class of hypotheses that suggests that various organelles in eukaryotic cell are descendants of endosymbionts, which are microbial side another microbial cell. Particularly, this descendant of endosymbionts are mainly the mitochondria and chloroplast. Already we have explained how this mitochondria and how this chloroplast are ingested into the cells. They are considered as descendants of endosymbionts. which are found in the modern eukaryotic cell. Mitochondria particularly evolved. It was hypothesized that mitochondria was particularly evolved from ancient aerobic or oxygen using bacteria. Chloroplasts evolved from photosynthetic bacteria or cyanobacterial category, specifically cyanobacterial category. Okay.
Thank you, sir.
Any other confusion? If not, I'm going to end this slide here. Now I'll go for the next. The next part of the syllabus is the molecular phylogeny.
Sir, can
Is
we
it
get this
visible?
PDF?
Yes, I can give you PDF, but again, I'm saying many of you they ask for the PDF. These PDF are for my reference. These are not sufficient. Don't write these things on your exam paper. Will not get mark, full mark. These are for my teaching, for my reference. But I want to give one person who can share to all. I can't give many of you they requested me for this PDF. Yes, I don't have any objection to give this PDF, but this is my request. Don't write these PDF points on your exam copy. These are for my reference. These are for my teaching. Okay. You have to elaborate it from different source. So many sources are available in the internet. You have to elaborate those sources because I am explaining each and every point in detail.
Sir, we have a group, so you can share anyone so that you can share in this group.
Okay. If anyone voluntarily can share the slides, then I can provide those slides to him or her.
Sir, I have already asked you to send the PPTs
Uh,
yesterday.
your good name, your good name, please.
Sir, in WhatsApp, my father name is given Akshay Kumar Patra.
Okay. So, are you interested to share the slides in the group?
Yes, sir. I have already in the group.
So, I'll share it by.
Sir, share the PPT in our group. It's good for all the students.
I'm not in your group. I am not in your group. That's why I can't share the PPT in your group. I can share it to one, and he or she has to share. So, yesterday, okay, we'll discuss it after the class. First of all, we'll discuss about molecular phylogeny. Under that, first we have to discuss a brief history and definition of this molecular phylogeny. The molecular phylogeny explains or refers to the study of evolutionary relationship among organisms using molecular sequences such as DNA, RNA, or protein. This is the definition for this molecular phylogeny. And this method was developed as a more objective approach to construct phylogenetic tree. Already we have discussed in the previous slide that for making a phylogenetic tree, we have to arrange them. We have to isolate these organisms in groups. Otherwise, we are unable to make a proper phylogenetic tree. Okay. So for construction of this phylogenetic tree, it is very important to study about their genetic material, to study about the protein, their synthesis, their replication pattern, their replication mechanism, everything. Yes. If we don't make them proper group, if we are unable to make them proper group, certain constraints are there, certain misleadings will come to our mind. What kind of misleadings? This convergent and divergent evolution. Some organisms, they have different structure but similar function. We can't make a proper group by using those properties. That's right. Just look at this picture. The convergent evolution, the wings of butterfly and the wings of bat. One is from mammalian group, one is from insect group. They are completely hell and heaven differences are there. Genetically, they are completely different, but their parts, wings of bat and wings of butterfly, they perform the same function. Okay, so to make a phylogenetic tree, to know about their ancestral group, we have to go for the genetic arrangement of the organism. Otherwise, it will be difficult for us to make proper phylogenetic tree, error-free phylogenetic tree. All of you clear? Why we are studying about their genetic material? Why we use particularly this ribosomal RNA, the DNA, the protein synthesis machinery to construct a genetic material? If I am audible, please say yes or no. If I am clear.
Yes, sir.
Yes,
Yes,
sir.
sir.
Yes, sir.
Let's come to certain key terms regarding this molecular phylogeny. What is phylogeny? It is evolutionary history of a species or group of related species. The molecular clock is the technique that uses mutation rate in DNA to estimate time when two species diverge. Okay, that means two species have common ancestor, but due course of time, due to mutation, due to environmental pressure, they diverge from each other. Okay, that mutation rate in DNA is estimated by using the molecular clock concept. Homology is the similarity in DNA sequence or gene due to shared ancestry. Orthology is the duplication within genome, while orthologs are genes in different species that evolved from a common ancestral gene by speciation. So here one example is there, paralogs and homologs or orthologs. Just go to the last paragraph, everyone. Go to the last paragraph. Last but one paragraph explain about homology. And the last paragraph explain about orthology. So this homology generally explain about the similarity between two traits like genes, like organs or structures, because every morphological characteristic is the expression of genetic characteristic. Yesterday I have explained. So this homology explain about the similarity between two traits. Try to understand it. What I am saying, listen it very carefully. These are actually the explanations. If you have rough note, then write it carefully and in a fast manner. Again, I am saying the homology means the similarity between two traits. We can consider these traits as genes. We can consider the traits as organs or structure, everything. What does it mean? It means that these organisms inherited or these traits inherited from a common ancestor. Okay, we can take one example, the four limbs. The four limbs of human, it's an organ. Here, trait is the organ. The four limb of human, whale, bat, cat. What is the arrangement of four limb? The arrangement of four limb is humerus, radius, ulna. Okay, but in different environment, they function in different manner. Whale have this kind of four limb. Human have this kind of four limb. Bat have this kind of four limb. The arrangement of humerus, radius, ulna. But whale is using that four limb in swimming. Bat is using that four limb in flying. Human is using that four limb for the purpose of food intake. Cheetah is using that four limb for running purpose. So these are the explanation of homology. On the contrast, if we go for the orthology, it is also a type of homology. That means when genes in different species are homologous but diverged after speciation. Again, I am saying genes in different species are homologous. Orthology explains what genes in different species are homologous but diverged after speciation, means new species formation. Also, we can take the example for this orthology. If we are consider the cytochrome C, the cytochrome C gene in human being, it's a either human being cytochrome C or mice cytochrome C. They are orthologous in nature. They are somehow similar in nature. Both help in cellular respiration, but they are present in different organisms. But they inherited from the common ancestor. Okay, so homology and orthology explains homology is the similarity due to common ancestry. Orthology is the homologous gene separated by speciation. In case of homology, we can give example of four limbs of mammal. In orthology, we can give the example of cytochrome C in human and mice. And the next part is the paralogy. Paralogy explains what? It explains the genes are homologous because they arose from gene duplication within the same species. Okay, we can take example. Example of hemoglobin and myoglobin. In human, the genes for hemoglobin and myoglobin are paralogous. Why they are paralogous? They came from duplication of ancestral gene. Got it? Hemoglobin carries oxygen in blood, whereas Myoglobin store oxygen in muscle. So when we compare this orthology and paralogy, orthology, they form by speciation, but paralogy, they form by gene duplication. This is the meaning of this paragraph. Okay? All of you clear about homology, orthology, and paralogy?
Yes, sir.
So what is gene duplication in a single word?
What is?
Gene duplication.
Yes. Your question is gene duplication. Duplication means simply the copy. When one gene make a copy of itself, that is the duplication. After duplication, two copies are formed. One copy keeps the original function, the another copy free to mutate. That's why change occurs. When we are considering hemoglobin and myoglobin, both are originated from the hemoglobin forming gene, but the myoglobin, due to mutation, they deviate from the hemoglobin nature. That concept is gene duplication concept.
Yes.
So certain limitations are there of this molecular phylogeny. What kind of limitations are there? Limitations means the drawbacks. At which point the molecular phylogeny is not accepted. The first one is the limitation of incomplete data or lack of complete data. Sequencing may not cover the entire genome, leading to incomplete data and misinterpretation. If we are comparing two genes and we are unable to get the proper genomic sequences, the proper genomic arrangement, okay, that is the phenomena which can mislead the phylogenetic tree formation. This is one limitation. Then horizontal gene transfer is another gene limitation. Why it is considered as another limitation? Because in case of bacteria, the horizontal gene transfer can obscure phylogenetic relationship by transforming genes between unrelated organisms. What does it mean when we are going to explain? It in a very simple manner, we can explain that this is the gene, horizontal gene transfer particularly, is the movement of gene from one organism to another organism, not by inheritance. If the transformation of gene by inheritance occurs, that is vertical gene transfer. But in case of horizontal gene transfer, genes move from one organism to another organism by horizontal manner, not by descendant. That means if one bacteria develops its sex phylus and another bacteria, they have the lack genetic material. through the sex phylus, one bacteria can transfer the genetic material to the next bacteria, which does not have that kind of genetic material. So, first one has a different ancestry. Second one, they have different ancestry. But due to this horizontal gene transfer, manipulation are there. And those manipulations are the component of misleading. Those manipulation can mislead the formation of proper phylogenetic tree. That's why this horizontal gene transfer is considered as one of the limitation for this molecular phylogeny.
Sir, horizontal gene transfer only occur in bacteria, or it is also occur in higher organism?
Generally, it is seen in lower group of organisms.
Okay.
Then convergence, the molecular sequence can evolve similarly in unrelated species, causing false relationship to be inferred. Okay, if same sequence, in certain cases we got the same sequence, then also that mislead the formation of tree, or then that can mislead to know about their common ancestry. Then computational complexity, so many codes are there. Analysis is very difficult. If any single mistake can cause a great harm to the phylogenetic tree formation process. So these all are some limitations to phylogeny or molecular phylogeny approach. But still, molecular phylogeny gives us idea about the ancestry. Though it is not 100% correct, but it is correct up to 100%, we can say that. That is the importance of this molecular phylogeny. So this is the process by which we can construct a phylogenetic tree by using molecular data. This is the process. So how to construct phylogenetic tree which illustrate the evolutionary relationship among the organisms? These are the steps, three important steps are there. First step, you must have to collect the data. So gather the data, the molecular sequence, data regarding RNA, data regarding DNA, its code, its replication mechanism, RNA synthesis, protein synthesis machinery, everything. So you have to collect the data. What is the data of different organisms you have to collect and gather first. Then second thing, you have to make a sequence alignment. That means align the sequence. If we are considering the genetic code, if this is organism A, this is organism B, this is organism C, first you have to make alignment of this organism A genetic code, then organism B genetic code, then organism C genetic code. Okay, so this is the alignment. And the next one is model selection. Choose an appropriate evolutionary model that describe how sequence change over time. That means up to which sequence they show similarities, and from which date, from which time, from which year they show the differences. And the difference point is the segregation point, bifurcation point from which evolution occurs. That might due to mutation, that might due to any other evolutionary force. So we have to make a proper model for that. This is tree building algorithm. What is tree building algorithm? distance-based method. So method use distance matrices based on sequence differences. That means how the genetic materials arranged, whether the genetic materials are very close to each other or the genetic materials are differ from each other. Then character-based method explains the maximum likelihood. Already I have explained the maximum likelihood means the maximum similarities found is considered as the ancestral property. If maximum similarities between two organisms are there, the first one is considered as the ancestor of the second one. Okay. And basing on those property, we can evaluate the phylogenetic tree. Just look at this diagrammatic presentation for selection of organism or gene family. Means we have to select different groups of organism or particular organismal gene for analysis. Then choose appropriate molecular markers, DNA, RNA, protein. Then go for its sequencing, its assembly, then make alignment. After alignment, make a proper model. Then, based on that model, we can make phylogenetic analysis. And on the basis of analysis, we can construct the tree for the purpose of evaluation. Okay. So this is the basic idea about the construction of phylogenetic tree using molecular data.
So molecular sequencing is done for entire DNA or a segment of DNA or gene.
No, this sequencing, if you consider three organisms or its genetic property, you have to choose that particular genetic property or the characteristic of that particular genetic property. Then only, if similar characteristics are compared, then only you can get a proper model. Otherwise, you can't get a proper model. Like one example, if organism A is there, you are analyzing the protein synthesis machinery of organism A, and organism B is there, you are analyzing the DNA sequencing of organism B. You can't compare these two things because these data are not identical data. So make identical data, then only you can compare. Okay.
So
Just
first traits should be considered.
yes, the trade should be considered. This is one example. If we are considering the nucleotide position of a particular gene or genome sequencing, then this is organism. This is for species A, the genome sequence is this one. Species B genome sequence is this one. Species C. Then only mark this color. You have to compare where you get maximum similarities. In case of 1, in case of 10, you got maximum similarities. Then you have to compare like this manner. Then only you can form a tree. Okay, this tree, this is the origin point. This origin point explains that certain organism, they completely different as they are the outgroup. Okay, and some of them, they converted to species A and species B. There is no evolutionary changes of species A, but the group which form the species B, they, due to environmental change or environmental pressure, they create another two groups like species C and species D. This we can analyze. This we can detect. This we can evaluate from formation of phylogenetic tree. Got it? This is the funny pictorial presentation for your understanding. All these groups, lamprey, sea bass, antelope, bald eagle, alligator, they all are different species. But some properties of them are common and some properties are different. Based on that property, we can make phylogenetic tree. But if we consider the external property, it is very difficult to make the phylogenetic tree. From the very beginning, I have explained that evolutionary constraints are there. Analogous and homologous characteristics are evolutionary constraints for making those phylogenetic trees. That's why for proper analysis, we have to go for genetic elaboration. We have to analyze the genetic elaboration for proper explanation, for proper construction of phylogenetic tree. All of you clear?
Yes, sir.
Yes, sir.
This is another example of phylogenetic tree. These are three domains, and under this domain, different kinds of organisms are there. This is the LUCA, last universal common ancestor, from which bacteria, archaea, eukarya are developed. These are different groups of bacteria. These are different groups of eukarya, archaea, and these are different groups of eukarya. Okay, so this is the phylogenetic tree topology. That means on this process we can separate the phylogenetic tree, the phylogenies. Root always explains us about what? About the ancestry, the common ancestry, the LUCA or the FUCA, first universal common ancestry or last universal common ancestry. Then internal node, branch, dichotomous branching, okay, terminal taxa. So these are different segregations based on different properties. So this is another important phenomenon, phylogenetic tree using 16S rRNA gene sequence. Okay, we all know that the prokaryotic ribosome, they have two subunits. They have 70S ribosome, but this 70S ribosome is divided into two subunits, one large subunit 50S and small subunit having 30S. Again, these 30S subunits have 16S subunit description. This 16S subunit, 16S rRNA gene sequence is used. How? For making the phylogenetic tree. We have to discuss in brief. The 16S rRNA gene is commonly used to construct phylogenetic tree, especially in prokaryotes. Okay, this is one of the example you have to describe according to your syllabus. This is the example you have to describe that make a phylogenetic tree by using 16S rRNA gene. It is not applicable for all the organisms. It is applicable for bacterial genome, and that we are discussing in brief in this class. Why this 16S rRNA gene is considered or is taking for consideration? Because the 16S rRNA gene is universally present in almost all the bacteria. and its evolution was very slow. These are the characteristics. Why we are taking this 16S rRNA gene? This slow evolution make it ideal for studying deep evolutionary relationship. If things are changing immediately, then it is very difficult to understand their property. It is very difficult to understand their behavior, their nature. But if the substance is changing in a slow manner, what is the process of evolution is explaining, then it is very easier for us to observe, then it is very easier for us to understand. Okay, that's why we are considering this 16S rRNA. We are taking this 16S rRNA as the base. Then another important property is conserved and variable regions in the gene allow comparison between closely and distantly related organisms. Okay, if the organism is closely related or the organism is distantly related, that also can be observed from the 16S rRNA position, okay, or analysis. This is the diagrammatic presentation of 16S rRNA. This is the part of 30S subunit, smaller subunit, okay, and this is the 16S part. One variable region is there, one conserved region is there. By using this variable region and conserved region, we can analyze the RNA sequencing. We can analyze this. We can isolate the RNA. Isolation process, RNA isolation process is there. By using that process, we can isolate that RNA, and after isolation, we can make sequencing. Okay. So what is the process of that? First, you have to amplify by PCR amplification for extraction of bacterial DNA. Okay. Extraction of bacterial DNA by PCR amplification. Amplify the 16S rRNA gene using PCR. I think all you know about the PCR. I'm just giving a very brief idea about PCR, polymerase chain reaction. Particularly, it's a lab technique which is used to make million copies of specific DNA sequence. Why it is important? It is important. PCR is important because it is used in DNA testing, disease detection, forensic research, cloning. Okay. So for the amplification, we are using the steps like denaturation, annealing, extension. All these are different steps for this PCR. These are not the part of discussion. That's why I have mentioned this in a brief manner. First, we have to extract the DNA, and then by amplification process, we can extract the 16S rRNA gene using PCR. Okay. After extraction, we can make a sequence of this product. PCR product means this is the 16S rRNA. PCR product means the 16S rRNA is the PCR product. Then only we can analyze the sequence, which is called as sequence alignment or the arrangement. Already the process we have discussed in previous slide. It is just the example. Okay. By using 16S rRNA, we can form a phylogenetic tree. Then tree construction by using distance-based method. This is distance-based method. By using this codes of 16S rRNA, we can segregate. Then after segregation, We have to construct the phylogenetic tree. So this is the process by which we can make phylogenetic tree. We can understand about the process of evolutionary ancestry, or which organism is the pure ancestor of which organism. All of you clear? Little bit clear about the phylogenetic tree, its construction?
Yes, sir.
Actually, the time is very limited. We have to cover many things within that short span of time. That's why we are moving a little bit faster manner. Otherwise, it is very difficult for me to cover all the chapters within this stipulated time. Okay, that's why I am touching all the parts as possible as I can. Okay, now the next part of your syllabus we are going to read, that is concept of speciation in bacteria. Concept of speciation in bacteria. Speciation means formation of new species. Got it? Here we will discuss the horizontal gene transfer. The pictorial presentation for horizontal gene transfer is there. Before some time, I have explained about the pilus. This is the pilus, through which one bacteria can give its gene to other bacteria that do not have that gene. Okay. If after some time we consider these two as single or common ancestry, then it will mislead for the formation of phylogenetic tree. Okay, so that part we have covered. Now come to speciation in bacteria. So bacterial speciation is the more complex than in multicellular organisms. Due to what? Due to horizontal gene transfer. Here, horizontal gene transfer explains what? The bacteria can acquire gene from unrelated species. This is another species, this is another species, completely two different species. But the horizontal gene transfer after some time make them similar, may not be same, but similar. That means the part of the gene which is not present earlier in the second bacteria, that might take from the first bacteria. Okay, this is one process. Another process is the recombination. The frequent genetic recombination can blur species boundaries. Genetic recombination means the aggregation. Okay, genetic recombination is the aggregation, after aggregation, exchange of genetic material occurs. Certain genetic material from one to another chromosome, or from another chromosome to one chromosome. That is the recombination process. And the operational taxonomic unit. In microbial ecology, bacterial species are often defined as operational taxonomy unit based on sequence similarity. Okay. As we discussed in 16S rRNA similarity threshold, almost all the bacteria, they have the 16S rRNA. That is the operational taxonomy unit. So what actually speciation explains? These are certain important terms that bacterial speciation is due to this concept. So what actually the speciation? Ecological speciation explain bacteria can diverge into distinct species through adaptation. to different ecological niche. This is the very important point regarding speciation. And for speciation, these things are required. If for this bacteria, F plasmid was not important in earlier environment, but the changing environment make a pressure for the requirement of this F plasmid. That's why that bacteria acquire F plasmid for its survivability. That is the ecological speciation. That that group of bacteria which was primitively lacked the F plasmid, now it has the F plasmid and its next generation they form F plasmid. Okay, so this is the bacterial divergence. The biological species concept is difficult to apply to bacteria, so the species are often defined based on genetic divergence and ecological factor. For smaller group of organism and for larger group of organism, for simpler organism, for complex organism, the species concept, the formation of species is different. Before some time, some of you asked about the horizontal gene transfer in complex organism. Just imagine if horizontal gene transfer occur in case of human being, what would happen? Human being might change to other species in the next morning if horizontal gene transfer occur, like bacteria that might survive, that might not survive by this technique. So biological species concept is difficult to apply in smaller group of organisms. Okay. Now the next part, the second unit is very big unit. The next part is your molecular divergence and molecular clock. Any confusion in horizontal gene transfer or bacterial speciation? If any confusion, ask me. We'll discuss it in 1 or 2 minutes. Please ask me if any question regarding horizontal gene transfer or bacterial speciation. Am I audible to all? If audible, then ask me if any doubt. Okay, if there is no doubt, then we'll go for molecular divergence and molecular clocks. First, we'll go for molecular divergence. What molecular divergence explains? Uh, it means the accumulation of differences. Here it is mentioned that the process by which genetic sequence in different species accumulate mutation over time, leading to genetic differences. This divergence form the basis of estimating evolutionary relationship and time scale. Okay, just look at this picture. This is a picture of human evolution. This is the origin. From this origin, chimpanzees were originated, Neanderthals were originated, and modern human beings also originated. So molecular divergence from past to present explains what? It explains about accumulation of differences. Differences means here what? Here differences are mutations. Mutations occur generally in DNA, RNA, or protein between two species or populations. Why this molecular divergence occurs? It occurs due to random mutation. Okay. For this random mutation property, mutation is the sudden change in genetic characteristics. For this random mutation property, the genetic material become more different from each other. Just look at this picture. When chimpanzees originated from the origin, and mutation at this point help in formation of these two species, Neanderthalis and modern human. So by course of time, more and more mutations occurs. Neanderthalis and modern human being, due to interbreeding property before 50,000 to 40,000 years.
Sir, I have a question.
Yes,
Sir, the random mutation occur in continuously or it is induced by the environment?
random.
Random mutation occur in continuous manner or it is induced by the environment? Due to environmental changes, the mutation occur or not?
Means you are asking about the causes of random mutation.
Yes, yes, sir.
Actually, random mutation is caused due to by chance. Okay, there is no specific purpose for random mutation. If we are taking some examples, so many factors are responsible for random mutation. Mostly the environmental factors are responsible for random mutation. When DNA replication is there, due to any kind of pressure, due to any kind of environmental pressure, if there is errors in replication, that may lead to substitution. Adenine is substituted by guanine. Okay, are you getting my point?
Yes, sir.
Spontaneous chemical change might be one of the factor. Radiation might be another factor. Chemical exposure, mobile genetic elements like transposons, everything we can take for the cause of random mutation. So random mutations, if accumulated over time, that is the cause for molecular divergence. Random mutation cause divergence in genetic material. That's why that make differences in organism. Okay.
Sir, if there is no pressure, the environmental pressure in the organism, there would be chances the less mutation.
There would be chances less means very less, negligible chance. If there is no change, why organism try to change itself? Okay.
Yes, sir. Yes, sir.
So these are the concept of molecular divergence. But the concept of molecular clock explains what? It explains genetic mutations accumulate at a relative constant rate over time for certain genes or proteins. Got it? By comparing molecular divergence between species and using known rate of mutation, the clock scientists can estimate when species diverge from common ancestor. It's a comparison, okay, between molecular divergence and molecular clock. Molecular divergence explain about the mutation in different time. If we are considering a particular segment of gene, if we are considering a mutation rate that is different in different time. But molecular clock concept explains certain mutations, they repeatedly occur due to particular environmental change, particular environmental pressure at a fixed period of time. So based on these two properties, scientists can evaluate an average time for the evolution of that organism. That's why these two things are very important. Calibration of molecular clock must be calibrated using fossil record. Fossil record explains about that by using carbon dating method, by using uranium lead dating method, we can collect the data. We can collect the data regarding the age of the fossil. So our molecular clock or divergence is correct or not correct, that can be calibrated by using that fossil record. Okay, if that fossil record is similar to our molecular clock data, then we can, our calculation is correct. Otherwise, our calculation is not correct. So many machineries are there in laboratory. They need calibration. If we are going for the blood test, different machines are there for blood testing for different parameters. But these machineries, the operators, the scientists, at a regular interval of time, they calibrate whether the machinery is showing best result or not, is showing the correct result or not. Okay, so these are particular techniques for calibrating our data, whether the data is correct or not correct. Okay, this is all about molecular divergence and molecular clocks. So these are certain pictorial presentation. Then come to the mechanism. What is actually the mechanism of molecular clock? Again, I'm saying the molecular clock explains about what? It explains about mutation at a fixed rate. Not all the mutations happen at a fixed rate. Fixed rate means the time is not exact, but time interval is almost similar. Okay, so what was the mechanism of this molecular clock? The rate of mutation in particular gene or genome can be thought of as a tick of the molecular clock. Rate of mutation is the backbone for this molecular clock. That's a tick. Tick mark means it is the point of reference. Some genes evolve faster than others with non-coding region or synonymous substitution. Okay. So mechanism of molecular clock actually it's the idea which works by assuming accumulation of mutation. Okay, whatever written here, its simplest version of explanation is it works by assuming that mutations accumulate in genetic material or protein sequencing at a roughly constant rate over time. Okay, already I have explained. The basic mechanism is random mutation happens at a steady rate in DNA over generation. This is the synonymous substitution. Here it is written synonymous substitution. It means the random mutation happens at a steady rate in DNA over generation. Then more time pass, more mutation accumulate. As we go with the time, they accumulate more. Here, just look at this picture. The time is increasing in this manner. That's why the organism at the top or this level are completely different from the original organism. Though the original organism is the ancestor, the final organisms are completely different from the original organism. That means more time pass, more mutations accumulate that make more speciation. By counting the number of differences here, by observing this, we can count the number of differences. By counting the number of differences between two species DNA, scientists can estimate how long ago they shared common ancestry. Okay, by using this time calculation from molecular clock. Time calculation means at which position, before how many years mutation occurs. Mutation occurs at a fixed rate means the last mutation occurred in this year. Last to last mutation occurs in this year. At which point, at which year, at which time the mutation occurs. By counting the number of differences. Differences means mutation. Repeatedly, I am saying by counting the number of differences, the number of mutations between two species, scientists can estimate how long or how long ago they shared a common ancestry. Okay, so this all about the mechanism of molecular clock. This neutral theory of molecular clock proposed by Moto Kimura. What this theory explains? It supports the idea that most evolutionary changes at the molecular level are neutral. That is, do not affect an organism's fitness and accumulate at a relative constant rate, providing the foundation for molecular clock. It is very interesting explanation. If mutation is occurring every day, every time, because every time there is environmental pressure, but we have a resistant mechanism, mutation resistant mechanism in our body. All the mutations are not the constant and fixed mutation in our body. Most of the mutations, they can move to its original condition. The technique is DNA repair technique in our body. Otherwise, it will be very difficult for us to survive without having the repair technique, without having the reverse mutation technique. We can't survive on the surface of Earth in the changing environment. Okay, so Moto Kimura explains that most evolutionary changes at the molecular level are neutral. That means, though mutation occur, the mutation become reversal. Okay, do not affect an organism's fitness. Fitness means survivability and reproductive capacity. If mutation occur in a frequent manner, organism unable to survive, organism unable to reproduce. That's why Moto Kimura explains that molecular evolution, most of the time, they show neutralism. Okay. Then come to the next part. Just a minute. This is molecular drive. It's the process by which genetic changes spread through a population, not necessarily by natural selection, but by mechanisms such as gene conversion, biased gene conversion, and unequal crossing over. Okay. Natural selection, in the previous class, we have discussed about natural selection, what it explains. It explains the presence of gene, if suitable, towards the natural condition, then only nature select those organisms. But molecular drive explains that genetic changes spread through a population, not necessarily by natural selection, but by mechanism of gene conversion, like not reciprocal transfer of genetic material between homologous sequence. Okay. Gene conversion, if we discuss about gene conversion, I'm just trying to explain it in a simple manner. Gene conversion explains about the DNA sequence when one DNA sequence copies itself over another similar sequence, making them more identical. It is called as gene conversion. Basic idea behind gene conversion is it's not a mutation, but a correction, overwriting event between homologous DNA sequence. It's not mutation sequence, it's a correction between homologous DNA sequence. How this conversion occurs? This conversion occurs during DNA repair. Okay, two similar sequence line up. And sometimes instead of exchanging bits evenly, one sequence is used as a template to correct or overwrite part of another. These are the reasons for gene conversion. That is essential for molecular drive. Next one, biased gene conversion. Biased gene conversion means a preference for certain nucleotide sequence to be inherited, which can drive certain genetic variant to fixation in a population. That means a particular gene in every generation is selected. That is the biased gene conversion. An unequal crossing over is during meiosis. Unequal recombination between chromosomes can lead to duplication or deletion of gene sequence. If duplication or deletion occurs, that contributing to genetic variation. So in concluding remark, we can say that this molecular drive can lead to a rapid spread of certain genetic changes within population, sometimes complicating the interpretation of evolutionary histories. Natural selection is not explaining about the rapid change generation after generation. It explains about the change in a slow manner, generation after generation. But this genetic variation, molecular drive explains that within a population, we can see mutations in a faster rate that can cause harm to the process of evolution, the process of fitness, or they interpret in the mechanism of evolution. So this all about the second unit concept. And in the second unit, we have covered many things only left part is divergence of bacterial and archaeal genome, origin and divergence of bacteria and archaea with bacterial genome evolution. Already from this part also we have discussed so many things. Horizontal gene transfer. Only we have to discuss about role of plasmid, transposon, integrons and genomic islands in DNA transfer. In the next class, we are going to discuss about these things. So from today's class, if any doubt, you can ask me whatever doubts you have. 5 to 6 minutes left. Now this is the time to clear some doubts, certain doubts. Okay, the house is open for question. Please ask. Am I audible?
So what is convergent evolution?
When we are talking about convergent evolution, if many organisms are there with different ancestry, means they derived from different ancestor, but in the recent time, they are performing the same function. Like in this example, think about the bat, think about the butterfly, think about the bird. They all have wings, but bat wings composition structural analysis is not similar as butterfly wing. Bird wing composition or structural analysis is not same as bat or butterfly, but their functional ability is same. It shows that though they have different ancestry, they converge at a particular time, at a particular period. That explains about the convergent evolution.
Divergent evolution, sir. Divergent evolution.
Divergent evolution means if they originated from a single point or common ancestry, but they differ from each other.
Thank you, sir.
Sir, what is molecular marker and its function?
Again,
What is molecular marker and its function?
molecular marker. Molecular marker, where we discussed about molecular marker. Okay, I think molecular marker is the specific DNA sequence, okay, which can be easily detected, and particularly it is used for genetic differences. Got it?
Example, sir?
Like identifying species. If we are taking the purpose of identifying species or strains, the example we can cite here is detecting plant varieties or bacterial types. For studying genetic diversity, we can take examples of checking how closely related two populations are. For disease diagnosis, also we can take the examples of finding genetic mutations, which type of mutation causing which type of disease. Okay.
Okay, sir. Any enzyme, any what thing which is used for molecular marker?
Not audible properly.
Which is used in molecular marker technique.
What is used?
No, sir. Any enzyme or chemicals which is used in the molecular marker technique.
Okay. We, for molecular marker, we can use the materials like specific DNA sequences, like unique part of the DNA that vary between individuals. Like RFPL. One example is RFPL, restriction fragment length polymerization. Okay, that we can take as example.
The restriction.
Restriction fragment RFPL. Restriction fragment length polymorphism. SSR also simple sequence repeat. SNP single nucleotide polymorphism. Okay, so these are the examples of molecular markers.
So what type of marker is used to differentiate between two different species?
What type of markers?
The species which belongs to same genre but they belongs to different species. What type of marker we can use to differentiate them?
I'm not getting it properly. Still, I'm trying to convince. Means, I think your
Sir,
question
sir,
is
there
depending
is
upon species. My,
sir, there
if
is
I'm
different
getting
kind
you,
of. Yes,
if
sir.
I'm getting you for my point of view, I'm just explaining. Try to understand. If I'm not wrong, you are asking me about depending on species, like different species, microorganisms, plants, animals. Scientists choose different markers. Is it your
So,
question?
like pangolin species, there is different pangolin species in India, in different region of India. Then how can I differentiate between the two pangolin um, belongs to different species?
When we're taking the common animal species, the microsatellites like SSR, mitochondrial DNA, SNP, they are the common markers, common used markers.
So that can be different. That can be different in within the species, no? In different individual.
Yes.
That the marker can also different in between two
Like
individual.
in human being, SNP, SPR, short tandem repeats, we can use as marker in human being.
Yes, sir.
Okay, have a good night to all. I'm going to stop.
Thank you,
Okay,
sir.
sir.
Hello,
Thank you.
hello
Today
sir.
we'll discuss the next part.
Hello sir, ଟିକେ PPT ପଠେଇବେ?
ଆଉ କାହାକୁ ଜଣକୁ ମୁଁ ପଠେଇବି ସେ ଯୋଉ ମତେ ଯିଏ କାଲି ଆଜି କହୁଥିଲେ ଯୋଉ ମାନେ ମ୍ୟାଡାମ ମୁଁ ତାଙ୍କୁ ପଠେଇ ଦଉଛି ସେ ଗ୍ରୁପ ରେ ଯଦି ସେଣ୍ଡ ନ କରିବେ ମୁଁ କାଲି ଆଉ ଜଣକୁ ପଠେଇବି ବଟ୍ ମୁଁ ଭାବୁଛି ତାଙ୍କୁ ପଠେଇଲେ ସେ ସେଣ୍ଡ କରିଦେବେ ମୁଁ ଆଜି ପିପିଟି ପଠେଇ ଦଉଛି
ଆଜ୍ଞା sir.
ଥ୍ୟାଙ୍କ୍ସ ଟୁ ଅଲ୍
Thanks sir.
Thank you, sir.